happy to introduce Dr. Andy Bader. His expertise spans early stage drug development to phase 1 clinical trials, specializing in oligonucleotide cellular therapies. In his academic career, he investigated cancer mechanisms, validating drug targets both in vitro and in vivo. As leader of the mRNA therapeutics program at Astrogen Inc., he directed research programs focused on microRNA-based therapies. Let's catch his engaging presentation on cell therapy for cancer.
All right. Thank you for having me. Pleasure to be here. I'm actually currently the CSO of a company where we develop novel cell therapies for cancer. Therefore, the title, but I changed the title just a tiny bit and added a path to it because I wanted to touch on a few things that parallel some aspects that were discussed earlier, particularly the microRNAs, which I was part of for more than 10 years developing therapeutic microRNAs, but I also had the pleasure to work with multiple different modalities over more than 2 decades. So that's why I'd like to reminisce a little bit about where we came from, what we have learned, and where we stand and all that today. So with that, in the spirit of this meeting, just a few stats, and we have seen a few more stats already about oncology in females. Interesting, everybody used a different resource, but in the US for females and indications like lung and breast, due to early detection and the involvement of surgery early on. But clinicians tell us that they expect an uptick of certain cancers within the next 15 to 20 years, particularly liver cancer, colon, and pancreas. But I'm not sure if they know the upswing of GLP-1 agonists, which was just mentioned previously, because they have a very strong impact on diet, and there's a lot of dietary related cancers, and actually that might, again, bring the incidence down. There have been already reports where GLP-1 agonists reduce the frequency of some of these cancers. In terms of a comparison, female between male, it turns out that the females fare slightly better. So it's interesting to see that the incidence across various cancer types is lower in females, sometimes substantially, sometimes minimally, but nevertheless. Breast cancer obviously is very prominent in females, which brings up the number again, which almost brings it up to 50%. Now, certain articles talk about why that might be. It might be due to longevity of females versus males, which is longer in females, and cancer is definitely positively correlated with age. But we also need to bear in mind that cancer is often presented in conjunction with other comorbidities. But something that is less talked about is that men just don't do a great job going to doctors. And so that might actually play into this with certain cancer types. Again, cancer and the outcome of cancers has all to do with early diagnosis and the stage of the disease. So we've seen a similar graph before. I just want to reiterate that, yes, breast new cases is the top winner, followed by lung, but not in the deaths. But breast is still a pretty prominent and scary disease because especially after cures, it can come back after a decade or more, for instance, in the brain where it's typically fatal. So there's definitely an unmet need. And I will talk a little bit later about a HER2-directed cell therapy program that we've been running. All right, just want to start a little bit now, a history lesson. Also, as part of my own personal journey, when I started more than 20 years ago, and then ended up doing a postdoc at the Scripps Research Institute in the early 2000s, the field was just about to shy away from traditional chemotherapy, try to do something that is safer since chemo affects normal tissues as well, and the therapeutic window can be sometimes very small. Targeted therapy came about and created some early promising successes in heme malignancies. For instance, inhibitors against the BCR-ABL oncogene that was fast-tracked approved in 2001 in CML. So around that time, Bernard Weinstein coined the term of oncogene addiction, meaning that if you identify that leg in the cancer and take it out, the entire cancer can collapse, and maybe that one leg is actually just one target, one oncogene. So he published in Science the promise of the Achilles heel of cancer. So mixing out the heel, but replacing one of the E's with an A, because that would be the remedy to all of that. Now, this has worked in hematological malignancies, and then also set an expectations for solid tumors. And so my work actually involved looking at the PI3 kinase pathway, which is an oncogene itself, tracing down the oncogenic signal to mTOR and others. And just like what you typically do, in drug development, you understand the cancer biology, you identify a therapeutic target, and that leads you to your therapeutic intervention. And we worked with, for instance, mTOR inhibitors like everolimus, which is also branded as Afinitor, which then gained approval by Novartis certain cancer types, multiple. It looked promising, but there remained challenges. And so just only from this graph, you see that we cannot speak of cures. So we essentially inhibited the cancer as long as we gave the drug, even in a preventative way, then the tumor actually flared up again. And so in the advent of cancer genome sequencing, much of this work done and led by Charles Swanton and then many others as well, we learned that intratumoral heterogeneity can challenge, if not even undermine, the concept of targeted therapy, because it's an ongoing evolution in your body, multiple biopsies taking at different places from metastatic lesions, but also the primary showed that genetic profile never really stays the same. It is probably a consequence of selective pressure that happens in your body that leads to then eventually more aggressive forms of cancers. So you can build these nice phylogenetic trees and actually connect the dots where the tumor arose and how it developed into. But what's more is case studies that trace cancer patients over time. For instance, in this case, 9 years in an AML patient that was first diagnosed and then put on different types of therapeutic regimens. After each, the patient relapsed. So here in red indicated 1R, 2R. This is the time when the patient relapsed. And what you can see is that each therapy had a very profound effect on certain clones within the tumor. However, other clones prevailed, then grew back, sometimes even became the dominating clone within the cancer. And so that actually raised a whole discussion in the field how cancer therapy may also have implications on cancer growth, because it enhances that selective pressure. And does it even make the tumor even more aggressive than in the first place needs to be? He also showed us and demonstrated to us that sometimes progression-free survival is oftentimes correlated the time it takes some of these clones to grow back and fill the niche. That really demanded better solutions, how more broadly we can really target cancer as a whole, because cancer is not one entity in our body, it's multiple, it's hundreds, if not even thousands. So this brings me back to what back in the day, again, we already considered as the Bible for cancer biologists, the hallmarks of cancer published by Hanahan and Weinberg. There's 6 criteria, which then has been increased to 10 to appreciate the role of the immune system as well. But he already demonstrated that there's a lot going on in cancer, which provides multiple avenues for therapeutic intervention. But again, it also demonstrated the complexity of the disease. And so when siRNAs came about in the 2000s, everything became druggable, which was really a phenomenal concept, because you can really choose the drug targets that you like and devise medicine against it, and even multiple. But a few years just after, we discovered the endogenous, naturally occurring counterparts of siRNAs, which are the microRNAs. And they also have been implicated in diseases, and specifically cancer. So for instance, depending on the microRNA, so for instance, if a microRNA functions as a tumor suppressor, it can be silenced by epigenetic changes within the DNA and no longer be expressed. Therefore, you lose that suppressive function and cancer can arise. One of these microRNAs was miR34. And what microRNAs, however, naturally do is that they regulate multiple genes and therefore multiple pathways within cells. They're really gatekeepers of entire functional processes. What we learned from miR34 is that miR-34 was a very classical tumor suppressor that regulated all kinds of different processes that are associated with these hallmarks of cancer: cell cycle, anti-apoptosis, metastases, and many others. So as I was part of Myrna Therapeutics, in fact, I started out at Assuragen, and here you see the timeline of miR-34 from discovery into becoming the first microRNA mimic ever tested in a clinical trial for cancer in 2013. I was brought on board to Assuragen to develop that therapeutic idea of microRNAs, create some interesting proof of concept data, and then spun out Myrna Therapeutics, where we really took this head on. But the point I really want to make is that it was at the time when microRNAs became this new universe of drug targets, and nobody really knew what the functions were. So we kind of had to figure out ourselves what microRNA to choose to take it forward as a therapeutic. Again, microRNAs can function pleiotropically. So some of the microRNAs that were in the mix were in some context pro-tumor, but then in some context anti-tumor, but we wanted a clean molecule. So, we did a lot of work, research on the side ourselves, which actually was fun. But we had it selected as part of our pipeline before it was well known as a direct target of p53. And then later on, and we did some of those studies as well, it appeared that miR-34 can elicit many of the functions that also p53 has. And so in some ways, we could again reiterate and reinstall the concept of p53 gene therapy for cancer. To date, and probably even later on now, there's probably more than 30 oncogenes that have been validated as direct targets of miR-34, which makes miR-34 a very attractive therapeutic entity. We've done a lot of preclinical studies to show that, yes, indeed, we can regress tumors in various models of cancer because of delivery and the way we deliver the oligo payload as a liposomal nanoparticles. We very much focused on hepatocellular carcinoma. So we also were better than the standard of care at the time, sorafenib, which is, in some ways a very toxic drug. So clinicians always ask us, yes, if there was a new form of therapeutic for liver cancer, then please let us test it. And so that's what we did. So we initiated a phase 1 study that turned out to be a very expansive clinical trial, simply because miR-34 worked in every single cancer indication you tested in preclinically. It was such a powerful tumor suppressor. And so we tested it in solid tumors, and within solid tumors, liver cancer patients that we escalated separately simply because the toxicity profile can be very different than in other solid tumor types. And we had a basket cohort with non-HCC indications, but also heme malignancies. And on top of that, we tested 2 different dose regimens at knowing exactly what will give us the better exposure. Tested the drug in 4-week cycles, where we gave the drug 3 weeks, twice a week, or we tested it in 3-week cycles, 3 times 5, meaning that we infused these nanoparticles into patients on 5 consecutive days and then waited 2 more weeks before we repeated that cycle. And so based on the pharmacokinetic profiles shown on the right, we actually saw that we get better exposures with a QD times 5 regimen as calculation of area under the curve. So that's what we went forward with, and we continued our trial there in solid tumors specifically. We had some interesting biomarker data and signs to show that the drug actually was active. Number one, not shown here, but had few biopsies from these tumor patients where we did see that the microRNAs made it into the tumor bed, and these were studies done by in situ hybridization, so we had direct evidence. But as a surrogate tissue, we also used white blood cells, VMCs. And what we saw was a dose-dependent downregulation of predicted and validated miR-34 targets. So once again, miR-34, the way they work, the repression on the mRNA level can be actually quite subtle. So to get statistical significance, it's much better to use an entire signature, because then the analysis become very, very powerful, and that's what we saw. Now, on the clinical front, in terms of efficacy, we also had some interesting efficacy in several of the patients. So in total, with this dose regimen, we ended up enrolling 85 patients. We had 3 PRs. So in the parentheses, you see the percentage of patients within the phase 1 cohorts. And we had 32 stable diseases. Some of the examples of the PRs are shown there. There were confirmed partial responses, meaning that the tumor size was reduced by 30% or more. Even in one patient, we actually had data before the onset of treatment, because oftentimes you want to see the trajectory before, after treatment. And if you change that curve, it could be a pretty good indication that something is happening in this patient. But overall, this all happened in 2016, which was not a great year for RNA therapeutics, unfortunately. The risk-benefit ratio didn't really justify going forward with this. And so the board only let us try this once, and we weren't able to follow up. But for this audience, I want to make the point that microRNAs, I think the biology is very exciting, but hasn't really harnessed yet therapeutically. I think there's still a lot of options how we can harness the biology, not only in cancer, but other diseases as well. But this brings me to cell therapy, because we still learned a lot from that experience. So cell therapy, a renaissance in 2012 and up, with, for instance, the example of Emily Whitehead shown here. She was the first one dosed with what's now known as Kymriah, which is a CD19-directed CAR T-cell therapy, and CAR stands for chimeric antigen receptor. It's a very personalized medicine. It's autologous, meaning, and this is shown here on the right, take blood cells from the cancer patient, isolate the T cells, then genetically, essentially it's a form of gene therapy, you induce a transgene stably, integrate that transgene into the genome of these T cells. The transgene encodes that CAR, and I will later explain a little bit more what it does. And then you expand these T cells ex vivo, have a ton, and infuse it back to the patient. So the outcome is twofold. One is, through this process, you make a lot more healthy T cells than the patient would ever have naturally, because oftentimes the immune system, specifically T cells, the function can be compromised. These patients oftentimes undergo chemotherapy, and they all have an impact on the viability and functionality of these T cells. But then second of it, you make these T cells, again, recognize and see tumor cells. So there's various ways how tumor cells can evade the immune system. One is by not expressing the targets that T cells would react to naturally. And so in this case, you lead the T cells to the CD19 surface marker, which is a highly selective cell surface marker for B cells. And yes, we would also address normal B cells. That actually can be clinically managed. So most of the patients that respond to these type of CD19 therapies also experience B-cell aplasia, but that's okay. So there's also another rationale for highlighting cell therapy, because in reality, it's the most dynamic drug that was ever invented. And we now know that cancer is a dynamic process within the body. And it's dynamic on a number of different fronts. So for instance, it can adapt to tumor masses, because T cells and other immune cells can naturally expand. And it's a response to the tumor in itself. So for instance, in mouse models, beautifully, the PK profile is an inverse correlation with tumor kinetics. So as long as there's tumor around, T cell will expand and attack the tumor, but if the tumor disappears, the T cells also disappear, they contract. And we also have now finally a way to address metastatic disease, which has been always a key challenge. And again, most cancer patients die of metastatic disease, not of a primary local tumor. These T cells, they seek out the tumor. They can directly home to the tumor, even the little tumors that you will never find in a PET or a CT scan. And then the holy grail, obviously, and this is more broadly speaking for immune oncology in general, is you reinvigorate the endogenous immune system. So essentially, break that environment of a cold tumor that essentially has become the system its own and lives side by side with the immune system, so that the immune system no longer attacks the tumor. Break that stasis, make the tumor hot again, so that now tumor cells again can go after other tumor antigens other than those that you would otherwise therapeutically address with your therapy. So for those that are not too familiar with the space, here are just a few stats, because there's a number of ways how you can create cell therapies for cancer. This is fairly recent data. Obviously, the T cells prevail because it's no doubt the most cytotoxic immune cell there is that can adapt and really proliferate very strongly. But there's other approaches out there like natural killer cells, and we have a few other cells up here too. For instance, in the mix that is not specifically called out, but there are also cells that use macrophages. Then dendritic cells, because the antigen presentation, which is facilitated by dendritic cells, is obviously very different too, so that the T cells can again see the tumor cells. Within the immune cell therapy space, it's highly dominated by applications targeting heme malignancies, and that is target specific. So CD19 is definitely the poster child that has borne many, many immune cell therapies, probably too many than it's really needed for this market. But among them, there are 347 others. And BCMA is another B cell specific antigen, which has had some successes in multiple myeloma. Then the question becomes, what do you do for solid tumors? Solid tumors, again, is a different beast than heme. Number one, genetically, it's a more diverse disease. It has a tumor microenvironment that is typically hostile and immune suppressive, which is not as prevalent in heme malignancies. And also, a little bit of a challenge for us as developers. Most of these antigens that you find on solid tumor cells are also expressed on normal tissues, because most of the time these solid tumors arise from normal somatic cells, and therefore they also carry these markers. So you need to find something that is more tumor selective. Within the cell therapy space, the CAR Ts prevail. So CAR is one way to engineer T cells with the synthetic receptors, but this is just one example out of many. It just had taken off because of the successes, but it may actually not be the best solution out there. Time will tell. And this is, I think, my last stats slide. I already hinted to that. All the successes we had to date in the CAR T space are limited to heme malignancy. So it is leukemia, B-cell leukemias, B-cell lymphomas, multiple myeloma. But solid tumors still remains an uncharted territory for the reasons that I just mentioned. So here's what we come in as Triumvira. So we developed a novel approach to tackle some of the limitations that we have seen with the CAR technologies. Let me explain a little bit what a CAR is. This is shown here on the right. I'll just back to it because it doesn't work. But essentially, it's a simplistic imitation of what the natural T cell receptor complex does. It's just that the natural TCR, these T cells, doesn't really work anymore because, for instance, it cannot find the antigen that would otherwise be expressed on immune cells, on tumor cells. So what folks have done is they have used a new antigen binding domain that recognizes a tumor, such as CD19, infused it to some elements of the natural TCR, and they also brought in maybe some co-stim factors to make the receptor a little bit more potent. But all the components are missing. What the receptor does is very effective redirecting of these T cells to tumor cells and turn T cells on once they see the antigen. What we also have learned is that you can actually turn T cells on when it doesn't see the antigen, i.e. the receptor is some degree leaky. That can have some detrimental effects to the T cells because T cells naturally have a shelf life. So you start with memory-like T cells, then once they get activated, differentiate into effector T cells, and then eventually will expire because that's the natural course that T cells will have. And that's essentially a safe mechanism to make sure that T cells don't over time overreact. What it also means is that if the receptor is leaky, it drives these T cells into premature exhaustion and therefore in some ways undermines the therapeutic potential that T cells can have in the first place. It also makes these receptors trigger happy, meaning that there might be more likely chances for off-target effects or even on-target effects in normal tissue. Okay? So great concept, it can work, but I think as a field, we also appreciate there's room for improvement. So this is what we have been doing. We actually have been building on the knowledge of CAR T data. We built a new receptor that we call the T cell antigen coupler, and simply as a different name because even though it's also a definition, a chimeric receptor, the definition of CAR and the abbreviation typically is reserved for this design that I just showed you. But the design that we made doesn't look anything like it. It's very different. It has an antigen binding domain. Then it has another domain that binds the natural TCR like a BiTE, but we added sequences of the CD4 co-receptor to it. And I'm sorry if I'm going a little bit deep, but here mechanisms really lies in the details. What we try to do is, and I hope this slide in some ways can illustrate this, is that we wanted to mimic the way natural T cells get activated because we appreciate that this receptor system is very complicated. It's actually one of the most complicated receptor systems that we have in the human body. And it is complicated because the T cell naturally needs to find a way to fine-tune the T cell response. Because we have this balance between no immune response, which is not good for cancer and fighting cancer, versus too much of immune response, which leads to autoimmune diseases. You don't want that scenario too, right? So what the CAR, for instance, doesn't have is naturally built-in negative feedback loops. Tell the T cell, okay, now there's a signal, kill the target, but once the target is killed, we stop signaling. That does two things. Number one is a better safety regulation because these T cells don't overreact. I should say a lot of these immune cell therapies are associated with toxicity profiles like cytokine release syndromes and neurotoxicity. We now know better how to manage, but if we didn't have them in the first place, I think that would be desirable. So T cells using that TCR typically have a way to auto-regulate themselves. Safety mechanism is better, but also efficacy, because once they stop signaling, they don't exhaust as quickly. What we have learned from essentially all cancer regimens is the durability of a drug regimen. Durability is key because we need to make sure that these T cells stay alive so that they can eradicate not only a part of the tumor, but the entire tumor. Again, this is the receptor that we have to date. On the right, we see what we call the quality attributes of T cells. And what we, again, set out to do is activate T cells in a normal fashion. So there's some cool signs that we can do via internal interference microscopy and look at how these T cells latch onto the target. Bottom line, it looks very much like a normal T cell. We know that our tag receptor is right there where the signaling occurs. This is just some proof of concept where we compared our therapy against standard CAR T using the same binder, in this case, a HER2 binder. And what I just told you in terms of a premise actually turned out to be true. We have much better efficacy. So in purple, we see the tag-treated T cells go against the tumor and completely reject it. Well, the CAR has some moderate activity, and that's fine. But the CAR suffered from toxicities. You see next to it the body weights of these mice. And what we have seen many times is actually that these CAR T cells at certain dose levels kill the mice, which prevents us from using CAR Ts at the dose that might actually lead to complete tumor clearance. We can't because of toxicity issues. On the right, it just shows you that these CAR Ts shed a lot more cytokines, again, in the spirit of what we know now clinically from CAR Ts, that they can induce cytokine release syndromes. I want to walk through this in the interest of time a little bit more quickly too, but there's a lot of data to corroborate this hypothesis preclinically. We now as a company have moved 3 products beyond the IND finish line. Each of these programs probably entails anywhere between 400 to 500 animals. So we're probably north of 1,000 animals, and in no case have we seen tech-related toxicities. But in most cases, we see complete tumor regression. Durability is key. This just shows that in preclinical models, we have great durability. One key thing about immune therapy, especially with cell therapy, is memory. So what we want to achieve is that once you give this therapy to your patient, it not only contains the tumor, but it also forms memory cells, just like with infectious disease, so that if the tumor were to recur, T cells can react very quickly and again contain the tumor. Preclinically, we actually do see that. So On the very right, you see an animal experiment where we have tagged T cells given to a mouse with an established tumor. The tumor disappears in purple, and then we give a new tumor after a prolonged time to make sure that T cells contract again, and there's enough of a gap between the first tumor and the second tumor. And in fact, we plan on the opposite flank as well. The T cells will have to migrate to a new tumor site. And what you see is a little bit of a blip of the tumor, but the T cells reject again. So no second dosing. It's just a single dose given at the beginning. So where are we today? We are in a phase 2 study right now with this HER2 lead program. This is the phase 1 portion of it that we published at ASCO of last year. Similar to cancer drugs, you have to escalate the dose, making sure that you administer the safe dose level and then go up to your therapeutic dose and define your recommended phase 2 dose. What we have seen over the course of 19 patients is, again, in agreement with our hypothesis, it's been very well tolerated. No neurotoxicity, which is a big win, because that's a pretty hefty side effect. And very few, only one grade 3 CRS. And most other toxicities were actually related to the lymphodepletion regimen, which is something that you commonly do to patients given a cell therapy because you have to create that niche for these other therapies to grow. The lymphodepletion in its own has a safety profile, and that's what we've been seeing in our trial as well. So here, just more of numbers and the data. You see the grade 1, grade 2, grade 3s, and so on. Most of them were lymphodepletion related. We had one dose-limiting toxicity, which turned out to be a pneumonitis. And just looking at the clinical profile, we think, we don't have direct evidence, but we think that could have been an on-target effect by our drug, because HER2 is also pretty prominently expressed just in normal lung tissue. Okay, in terms of efficacy, this is our waterfall plot across the different patients. So we have an 83% disease control rate, 33% overall response rate. We have a number of stable diseases, but also 2 partial responses, of which one led to the complete regression of the target lesion. And clinicians tell us that this is not what they're seeing in this patient population. So remember, this is a patient population that has seen 6 to 12 prior therapies. Life expectancy is very, very short. You typically don't see this type of responses in this patient population. So clinicians remain excited. We also learned, and again, coming to the theme of this conference, looked at breast cancer patients, we looked at other cancer types. What we have noted actually was that there was more of a response in patients with gastric cancer, and we don't really know exactly why. It may have to do with trafficking, because it is about getting the drug to the appropriate site where the cancer is, but these are also metastatic lesions, so there might be something that is more specific to gastric cancer as a whole. In terms of creating chemokine gradients and calling those T cells into the tumor bed, we don't know yet. So this really leads me to my last slide. So we as a company have recently press released the initiation of the phase 2 portion of the HER2 trial, with the main focus now on gastric cancer and gastroesophageal. We also have just now launched a second trial that we're really excited about against Claudin 18.2, which is a fairly tumor-selective marker for gastric cancer, because in normal tissues, it's actually hidden away in places where T cells cannot see it. But that expression goes awry, and it's vastly more upregulated in all kinds of different places on gastric tumor cells. And I just learned while I'm here that we dosed our first patient yesterday, so fingers crossed. Thank you. With this, I'd just like to close. Thank various parties involved in all this work, including you. Let me know if you have questions.
Thank you so much, Dr. Bader. We'll open it up to questions now.
Excellent talk. Really enjoyed it. So do I understand it correctly that your CAR T actually includes signal 1 and signal 2?
No, it's actually just signal 1. We don't need signal 2. So signal 2 was added to CARs because signal 1 was not enough to drive a response. That may have to do with the fact that they only use the CD3 epsilon, the CD3 zeta domain of the TCR. But as I showed you, there are more items in the natural TCR. We know that the signal strength is actually guided by the natural TCR. That's what we're doing. And we added the co-receptor domain, which, by the way, is not a signal 2, right? But the CD4 co-receptor domain is the most effective recruiter of LCK. The LCK is the initiating kinase for getting the signaling going, including CARs too. But now we know that, for instance, the CD28 domain within the CAR is less potent than the co-receptor domains of CD4. We think that's really critical in our design. No signal 2. Also, in some ways, you would not need necessarily signal 2 because these TAC T cells or any CAR T, they already have signal 1 once through the expansion phase, right? And then they are counter antigen again.
Okay.
But it definitely helps in the context of CARs.
Then the related question, what is the persistence of these T cells, and how would you compare, let's say, if you just stimulate naturally CD3, CD28?
Yes.
And somewhat related question, if you know it is the best type of T cells that has to transduce.
Yes, we transduce CD4s and CD8s, and we have a healthy combination of both. Also have a healthy combination of memory versus effector cells. Oftentimes memory is around 60 to 80%. We definitely think that you need to have effector, especially effector memory and TCM present to get the process started. In terms of durability, we don't know yet clinically. We have a PK profile where we see some sustained persistence of T cells, but it does drop off after day 29, which is when we take our first scan, which is why we also redose patients now. And in fact, the Claudin 18.2 trial, we're the first company where the FDA allows redosing as part of the phase 1 escalation, which is exciting for us. But preclinically, I can answer your question directly because, and not shown here head to head, this is what we call a repeat killing assay or recursive killing assay. So start with your CAR T or TAC T cells here at the very beginning, and you run them through rounds of co-cultures with your cancer cells. It lasts about 3 or 4 days long. And so you see that the cancer, the T cells very effectively kill target cells. So you harvest the T cells again and expose them to a fresh load of cancer cells. And you do this as long as the T cells can keep going. We can keep going for 48 days over 12 rounds. For a CAR, just to give you an example, they would drop off around 4 or 5.
Thank you so much, Dr. Bader.
Thank you.